Alright guys, so in this video we're going to talk about the kind of SRY gene on the Y chromosome. It's one of those pathways where, again, you can kind of put some there's little points in it where you say, okay, if I had a deficiency here, this would lead to this kind of presentation. So it's just one of those that I thought you could definitely use a refresher on, per se. So hope you like the video.
Alright guys, so here's the question. It says, a fetus with 46XY genotype has testes with normal functioning Leydig cells, but not the presence of Sertoli cells. Which of the following expressions is most likely to be seen? And you can see all the different answer choices. We'll talk about like male internal genitalia, female external, male and female internal, etc. So there's every combination. So you're probably going to see a question like this, and it may come under embryology or and such, but to get them right, you got to know the pathway that you see, and this is in your Step 1 book, but it's the one that talks about the SRY gene on the Y chromosome, okay? And so we'll put that guy right there, and you know, it's a guy. The guys have the XY, and the ladies have the XX. So we're talking the XY. So it's the SRY gene on the Y chromosome that produces testes differentiating factor. Okay, TDF we'll call it, and then that, of course, development of testes. So from there, and this is where you got to start memorizing this thing a little bit. From there, you're going to make the Sertoli cells, okay, and or the Leydig cells. Okay, so again, SRY gene on the Y chromosome makes the testes differentiating factor, which development of the testes. From there, you're going to have, they'll produce the Sertoli cells or the Leydig cells. Now, the Sertoli cells is going to make this thing called Mullerian. Okay, Mullerian, also we can kind of do an AKA, paramesonephrine, but Mullerian inhibitory factor. So it's going to produce an inhibitory factor. So if you do nothing, if you didn't have this, what does it inhibit from making? Well, this suppresses the paramesonephrine duct, okay, which basically makes the female internal genitalia, and we're talking, fallopian tube, uterus, and upper 1/3 of the vagina. Okay, so the Mullerian, if you didn't have this Mullerian inhibitory factor, the body would default to make this stuff. Okay, but since it's a guy, you need the Sertoli cells to make this Mullerian inhibitory factor, because it'll prevent this stuff from forming. Okay. And then over here, you're going to have the Leydig cells. Now, they're going to produce testosterone. Okay? And then testosterone, in the presence of 5-alpha reductase, is going to make a little more potent version of DHT. Okay? And then the DHT, we need that, because we put a little bit of that with the genital tubercle and urogenital sinus. So this, in the presence of DHT, will make the male external genitalia. Okay? So all you have to do is all you got to do here is memorize what's made, and then eventually we can go back and figure out where the problem is, and then you can understand how the person came to be like that. So male external genitalia, and prostate, okay? And so the last little piece of this is going to be testosterone, okay? It's called the Wolffian duct, okay? Wolffian, also known as mesonephric, in the presence of testosterone, testosterone stimulates Wolffian duct, and it's going to make the male internal, okay, internal genitalia. Okay, and that's going to be the epididymis, if I can spell it right, and vas deferens. Okay, well, seminal vesicle, etc., but not, all right, not the prostate, because we just talked about the prostate. Prostate's going to be part of this deal down here, where DHT, in the presence of the genital tubercle and urogenital sinus, it's that step that makes the prostate and such. Okay, so this is the pathway that you got to know. And again, this is a male, because, you know, without this, the default setting would be for a female. Okay, so that's the default setting, and everything just kind of, you know, produces into the into the internal female genitalia and such. But if we have an XY chromosome, on the Y chromosome, we can have this SRY gene, which will make the testes differentiating factor, makes the development of testes. That gives us the sertoli cells and the Leydig cells. And then you're going to know that sertoli makes the Mullerian inhibitory factor, and we need that if we're a guy, because the Mullerian inhibitory factor suppresses the paramesonephric duct, which therefore they don't form the fallopian tubes, etc., etc. You don't want that, obviously. You know, you're a guy. So, and then over here, the Leydig cells are the ones responsible for testosterone in the presence of 5-alpha reductase, can make DHT, and that, going through the genital tubercle and urogenital sinus, can make your male external genitalia. And then the Wolffian duct up here, in the presence of testosterone, made the male internal. Okay, so you really just have to know all this internal for the female stuff here, and then over here we're talking male internal, male external. Know where these are at, because here's where the issues come in. If I were to inhibit this guy, okay, 5-alpha reductase, for some reason, if I had a deficiency of 5-alpha reductase, which is known, it's actually autosomal dominant. It's, you know, they say it's sex. Well, I don't say sex linked. I got to make sure I get the right terminology on that. I think it's like sex associated. Sex limited is the terminology that they use. It is autosomal dominant. Sex limited to males. Okay, sex limited males, obviously, because here we are in this little pathway for males. And then, you know, with that, if I did not have this 5-alpha reductase, am I going to have any issues with the male internal genitalia? The answer is no. It's not going to affect that, because all I need for the male internal is just testosterone right here. Okay, but am I going to be able to produce the DHT? No, because I don't have this guy, or I got some type of deficiency of him. So, am I going to be able to make the male external genitalia? No, not like I should, right? So if I have a 5-alpha reductase deficiency, you know, I'm going to have ambiguous genitalia until puberty, because once the guy hits puberty, then he'll produce some extra testosterone, which will help kind of produce the male external genitalia when he's a little bit older. Therefore, when you have the 5-alpha reductase issue, you're not going to have the male external genitalia until later on. If I were to inhibit it right here, what would happen? Yeah, I'd be a male presentation, but what would be kind of the crazy part here is, like, everything over here is fine. This pathway is fine. So if I'm a male and I have an issue right here where I don't have any sertoli cells, well, I'll have normal internal male genitalia. I have normal male external genitalia. But what's going to be my problem? My problem is I don't have the sertoli cells to produce the Mullerian inhibitory factor, and therefore, what's going to happen? Well, I'm going to develop some female internal, genitalia. So I'm going to have some fallopian tubes, uterus, upper third of the vagina, so a little bit of a mix that we shouldn't have. So if you know this pathway and you know how it works on the SRY gene and the Y chromosome, it doesn't matter where they tell you the problem is or how the person presents. You can work backwards and say, well, look, I know if I have you know, if I have a problem with the male external genitalia, but everything else looks pretty normal, then I know it's probably a 5-alpha reductase. And later on, you know, when the testosterone levels rise, then things will look a little bit, obviously a little bit more normal. So now back to this question. It says a fetus with 46XY genotype, okay, has testes with normal functioning Leydig cells, but not the presence of sertoli cells. So right there, you know, I'm thinking, okay, SRY gene, it went down, you know, it made the sertoli here, and it made the Leydig cell here, and I know this was the guy, Mullerian inhibitory factor. Over here, he was messing with testosterone and DHT, which led to that's the external male, and then over here, we got the internal male genitalia. But the problem was there's no presence of sertoli cells, so we knocked this out. So what? I didn't get the Mullerian inhibitory factor. So what am I going to see? I'm going to see some female internal genitalia, right? That's going to be the fallopian tubes, uterus, upper third of the vagina. So not the presence of sertolis. Boom, that's out. So I got this. All this is normal, so this should be normal. And then this guy's sitting there because he wasn't eliminated. So is it A, male internal genitalia and female external genitalia? No, because I don't like that word external for the female, so that guy's off the board. Is it male and female internal genitalia? Okay, female internal, male internal with male external. Okay, looking pretty good. I like that one. Male internal and external genitalia. Yeah, but you know it's kind of true. But what are they missing here? They're missing the whole point of what the sertoli cell absence of that did. So I don't like that answer. There's obviously a better one sitting there so far. Or is it female internal genitalia? Yes, and male external genitalia only. Well, it's kind of true, but I can't say the only because I have male internal, so there's a better one out there. Plus, I don't like that really one, or none of the above. No, it's not that. The only answer here is going to be male and female internal genitalia, and then male external. Okay, so you got to go back and you really have to understand that pathway about the sertolis and the Leydig, and really what they perform. Okay, it's just a little bit of a strict memorization there, but it's something that's necessary on the STEP exam. And so this one says, an infant born to a 25-year-old female has ambiguous genitalia. There is presence of Y chromosome. Okay, so we're XY. So I'm thinking, whenever I see that, I'm thinking SRY gene, and then I go up here and I'm thinking sertoli, or I'm thinking Leydig, and again, testosterone, DHT, external male, internal male, Mullerian inhibitory factor, and then that's going to, if I need that, so I don't produce, that suppresses the female internal, okay? So there is no presence of uterus on exam. So I got a male, and there is no presence of uterus on exam, but they have ambiguous genitalia, no presence of uterus. Which of the following, this one probably should have said there is uterus, right? Which of the following is most responsible for the absence? Okay, there is, okay, so it was written right. Which of the following is responsible for the absence of uterus? So a lot of smoke and mirrors here, right? They put this ambiguous. Heck, they even, it was almost even, I got confused for a second. It's ambiguous, but there's no, the real question here is, which of the following is most responsible for the absence of uterus? Well, we know the absence of a uterus, especially when it comes to a male, is going to be due to the Mullerian inhibitory factor. Okay? So, again, when I wrote this, I should have known what's the real question here. This is the real question. All this is kind of smoke and mirrors. Okay? Now, some other stuff that you kind of just have to be aware of is that androgen insensitivity, okay? And basically on that is their phenotype, basically they look like a female, okay? And then also they don't have a cycle or something, and they finally go to the doctor. But, so they're a normal appearing female, and there might be like they call it like a rudimentary vagina, but the uterus is absent, okay? So you're thinking, well, what's going on here? But their genetic makeup is XY. So you have a person who looks female, like legit, and then their chromosome makeup's XY. The uterus is absent. But here's the key to this. Here's the key. There's no sexual hair, okay? Because what that's telling me is what makes the hair? The hair is made by androgens, okay? And if they're an XY and they're having massive failure when it comes to all their androgens, this is going to be the androgen insensitivity. And it's the fact that there's no sexual hair is what really differentiates this from anything else that may give you this, this kind of presentation where it's an XY and we're not really sure. But if you see where they have no sexual hair, it's an androgen issue, and that's with testosterone. So then there's an androgen insensitivity. And, with that, the testes, they say, are usually found in the labia, okay? And so what happened is the testes, when they descend, they go I had to look this up, transabdominal due to the Mullerian inhibitory factor, but then they descend into the testes. They descend into the scrotum due to testosterone. Okay, so what happens is even with this, that's just a kind of a what you need to know of how they kind of come down. So again, transabdominal is due to Mullerian inhibitory factor, and they descend in the scrotum due to with testosterone. Okay, some other terms that you might actually come across, and this is in your Step 1 book, is the Pseudohermaphroditism. I can't spell that. And really, all that really means is there's a disagreement, like it says, between what you see, which is the external genitalia, and genetic makeup, okay? So with a female pseudohermaphrodite, okay, they're an XX, right, because they're female. So you have ovaries present, but the external genitalia look, they call it virilized, not totally like a female, but it could be like a male. And now we can see that with what? The congenital adrenal hyperplasia. Okay? And remember that? We talked about that. We said, we had the deficiencies. So if we had like a 21 or 11 hydroxylase deficiency, it pushes everything to the sex chromosomes, or to the adrenal gland sex hormones. And so that would push it that way. And then that would give us a female pseudohermaphroditic presentation, which would be seen in congenital adrenal hyperplasia. Now, with the male pseudohermaphrodite, it's an XY because it's male. Testes are present, okay, but the genitalia is ambiguous, and we just talked about that, right? And we can see that with androgen insensitivity, okay? Insensitivity. And there's like varying stages of that. So this term here is almost used to describe, and then we can take it one step further and say, an example of female pseudohermaphroditism, where it's a female yet it looks more like a male per se, is going to be the congenital adrenal hyperplasia. The male pseudohermaphroditism is going to be a male, but it looks the opposite, and we can see that in androgen insensitivity. Okay, So just going back and looking at that one more time, you got to know this pathway. SRY gene on the Y chromosome makes sertoli and Leydig. Sertoli goes down, makes mullary inhibitory factor, which would inhibit the making of the female internal genitalia for a male. The Leydig makes testosterone in the presence of 5-alpha reductase. And then what medication treats that? Would we say finasteride would be actually like an inhibitor. It inhibits the 5-alpha reductase, therefore you get less of this, and that's why it can treat BPH and stuff like that. Okay, but the DHT in the presence of the genital tubercle and urogenital sinus can make the male external, which is going to be the penis prostate, and then the Wolffian duct in the presence of testosterone can make the male internal. So you got to know this pathway, guys. So hope this was helpful.